it's like, yes, look at my words, it's going at your eyes, and why is it here?
I think that last question was one where you needed to know about transporters. So there's your clue if you want to think about whether you would have known the answer. Any questions about thyroid hormone? Sorry. So you know what happened? Why I didn't get access to that Kahoot last time? I forgot to hit save after I'd composed it. Rookie mistake, right? Oh, well. Okay. So sorry it was out of sequence, but now we're going to go back to talking about sexual differentiation. And I won't go over this diagram again, except to say that in mammals, early in fetal development, all the sexes all look alike from the outside. Where they differ is that individuals are going to be male, typically have a Y chromosome, which has that crucial SRY gene on it. The presence of the SRY gene will cause the indifferent gonad to start developing into testes. In the absence of the SRY gene, the indifferent gonad will develop as an ovary. And from that point on, we don't know that the ovaries put out much hormone, but testes do. And the testes will release two hormones that masculinize the body. One is Müllerian regression factor, which is hormone, anti-Müllerian hormone. It's gone through several names over the years. That's the current one. Anti-Müllerian hormone, which is a protein hormone that acts locally to suppress the Müllerian ducts and therefore no oviduct and uterus and inner vagina form. And testosterone, which acts on the Wolffian ducts and causes them to develop in the epididymis, the vas deferens, and the seminal vesicles. In the absence of testes, in the absence of anti-Müllerian hormone, the Müllerian ducts develop into the oviduct, the uterus, and the inner vagina. And in the absence of testosterone, the Wolffian ducts wither away. And then the same hormones, I should say, testosterone is also going to be important in sculpting the outside of the fetus, so that in the presence of testosterone, the epidermal material between the two legs will develop a scrotum, and the phallus that is there in both sexes will develop more and therefore be larger and therefore be what we call a penis. And in the absence of testosterone, the tissues that are programmed to form labia and a clitoris and the opening of the vagina. And we'd gotten as far as pointing out that evidence that the exterior of the body in humans is totally controlled by the androgens like testosterone came from individuals with androgen insensitivity syndrome, where it's an XY individual. It turns out androgen insensitivity syndrome can only happen in XY individuals because, by coincidence, the gene for the androgen receptor is on the X chromosome. Okay? And that means that any individual with two X chromosomes will have at least one functioning androgen receptor. If you want to know why you can't, through ordinary breeding, get two X chromosomes with a defective androgen receptor in the same individual, I'll explain it to you later. Okay? But that means that an individual who has only one X chromosome, if the androgen receptor gene is defective there, then they will, the Y chromosome will cause anti-Müllerian hormone to be, the Y chromosome will cause the indifferent gonad to develop as testes. The testes will release anti-Müllerian hormone to suppress Müllerian duct structures. The testosterone would normally act on the Wolffian ducts, but they don't have the androgen receptors to respond to it, and so they fade away just as they do in females. And the periphery, which under testosterone's tutelage would normally form a male phenotype, instead forms a female phenotype. Everyone have that so far? And these individuals, in cases, there's a spectrum of androgen sensitivity. Sometimes it's partial androgen insensitivity syndrome, and that's one of the things that's causing problems in Olympic races these days in terms of deciding who's a female. But in cases where the androgen insensitivity is complete, in those cases, the individuals look totally like other females. Here's an example. These are all women with androgen insensitivity syndrome. I got to say AIS. With AIS, who meet annually as a support group. And you can see they look like women. This is the time where I will say that in this class and in hormones and human sexuality, normally for many years at this point, I would show pictures of an individual with AIS who was naked. Just as when I talk about male syndromes, I'd show pictures of such men naked. And you've seen such pictures probably, where the individual is shown naked, and typically there's a black bar across their face and to protect their identity. But then I learned that many of the people whose pictures have been taken, such as that, had no idea how widely they might circulate. And when you ask them, well, don't you feel better about having a black bar across your face, like you're a convict or a felon or something? Well, unsurprisingly, no. And so I don't show those pictures anymore. And I don't include them in my textbooks anymore. Because, I mean, what's the point of looking at a naked woman? She looks like a naked woman. You know what naked women look like, right? So here I can get across just as well. These people are women in every way. And as a preview of what we're going to talk about later, not always, but almost always, they are attracted to men. So they grow up being raised as girls. In many cases, they don't know that they carry an XY karyotype, don't know that they have internalized testes until puberty comes and they never have menstruation happening. How's that so far? And just to let you know that there is progress, even in medicine, if you're worrying about that. Once upon a time, OBGYNs, who every OBGYN practice would run into such individuals in the course of their practice, right? Because they're not that uncommon. One thing I'll point out to you is that the fact that individuals with totally no response to androgen grow up to be healthy and happy proves to you that you don't need an androgen receptor to live, right? Because they're doing just fine. The other thing that's interesting is until maybe 25 years ago, physicians were taught how to identify such individuals. So short vagina, shallow vagina, no cervix, right? Because AMH did its stuff. High levels of testosterone, typically higher than normal males. If you do a karyotype, you find a Y chromosome. At that point, you know what you're dealing with. And at that point, they were trained to start lying to the person, to never tell them what their syndrome was, to break to them the sad news that they would never be able to have children themselves, and then to lie to them and say that they needed abdominal surgery. And there were various pretexts given as to why to do the abdominal surgery. And the reason they needed to do the abdominal surgery was it was thought that because the testes remain inside the abdomen, they are more likely to develop testicular cancer. And so physicians were trained to lie to the person and tell them, you're never going to have a period, you're not going to be able to bear children, but everything else about your life can be just great, and we got to do emergency surgery to take out the testes. Several things were wrong with this. Number one is lying, which you don't understand. In the paternalistic model of medicine that dominated the United States till practically the end of the 20th century, it was perfectly commonplace and considered ethical to lie to your patients for their own good. The other thing is that while it is true technically that having testes remain in the abdomen make them more susceptible to testicular cancer, the total risk is very, very low. These days, I'm happy to say, slowly, practice is changing where they are telling these patients the whole story. Question about that? Although I will say it was only the patients who caused that to come about. Because what happened was there were more and more women with these being told this by doctors, and then the internet came and they went online and eventually figured out what was really going on. And do you think they were happy that their doctor lied to them for their sake? No, they were pissed. And then, it was only because of their agitation that questions about the ethics of that started to be raised. Justin?
People with complete androgen insensitivity, are they like hyperstimulated?
In many ways, they are. Yeah. I haven't talked about sexual differentiation of breasts, externalized breasts, because it's such a human-specific thing. But it turns out that the development of breasts is due to a ratio of estrogens and androgens. Okay? So if the ratio of estrogen is higher than the ratio of testosterone, then externalized breasts will develop. And if it isn't higher, then they won't. Well, these women have testosterone, right? Which, among other things, means, well, they have testosterone and they have estrogens that are also derived from both the adrenal gland and their testes. But because the androgen receptors don't respond, the net effect is a very high ratio of estrogen to androgen. Get that? And so, so they often tend to be very feminine. There are a few women, didn't present pictures of this, who have publicly identified themselves as having AIS, and I think that's pretty courageous of them. There's a memoir written by one, and I'm sorry, I can't remember what it is. I'll try to look it up, but it's very well written and spunky and totally admirable. Questions about that? Okay. So androgen receptors are important for the masculinization of the body. And starting back in 1959, people started realizing that early exposure to testosterone might also be masculinizing the brain. And these experiments were of this sort, where you would look at that feminine behavior, lordosis, that we showed way back here, the lordosis posture, and took that as an indicator of whether the brain was masculine or feminine. I told you that if you give a female mammal the right treatment with estrogens and progesterone, for rats and mice, it's a couple days of estrogen, then 1 day of progesterone, and 6 hours later, the animal will become receptive. What I didn't tell you is you can give that exact same hormone regimen to male rats, and they don't show lordosis. And if you want to see an example of this, well, hold on. And so what people found was that if you castrate males on the day of birth, then when they grow up and give them those hormones, they will show lordosis. So they concluded that something about having testosterone early in life changed the brain so that the animals would not show lordosis. Conversely, what they found is that if they provided females with testosterone, either before birth or right after birth, in adulthood, even if you gave them those estrogens and progesterone, they would never show lordosis. And contrary-wise, they were much more interested in mounting other females, whereas males castrated at birth are much less interested in mounting females. Yeah.
Castrated later on, they show.
If you wait until about like the second week of life to do the castration, it's too late. So it's a great question. And for that sort of behavior, people started defining a sensitive period during which if the brain is exposed to androgens at this period, then there'll be a change in their behavior. And the people who first formulated this idea called it the organizational hypothesis, and they proposed that hormones affect behavior in two fundamentally different ways. First, there's what happens when you give hormones in adulthood, where the animal's behavior is temporarily affected. And they said, in those cases, the hormone is activating the behavior. But they said when animals are given hormone early in life, there's a fundamentally different thing that happens. Exposure to testosterone early in life, they proposed, permanently altered the brain so that the animal's behavior was permanently altered. And they called this the organizational hypothesis, well aware that that's basically the picture that had come out in studies of sexual differentiation of the periphery in mammals. So they said, just as testosterone early in life organizes the body to take on a male phenotype, testosterone early in life organizes the brain to take on a male phenotype. And we can measure the brain's phenotype either by looking at lordosis behaviors or mounting behavior, and that'll give us a readout of how masculine or feminine their brain is. How's that? And I have a slide here to show you all that, what I just told you. And so I'm going to skip ahead a little bit to say that first I'll just say this business of having one hormone determine this sexual phenotype of the body, I hope you can see there's something kind of elegant about that, right? Just one factor that changes lots of different parts of the body, all of them coordinated to the same end. That is, let's figure out how to successfully reproduce as a male. And so there's something elegant about that, right? It's harder for things to go wrong if there's just one trigger. Things still do, of course, like everything in biology. But you can see, how that takes care of the problem of not making an individual in which, part of the body is masculine and part is feminine, and therefore they can't reproduce as a male or a female. And so the organizational hypothesis was similar in terms of its elegance, right? A Y chromosome determines whether there'll be testes. The testes will produce testosterone. The testosterone will masculinize the exterior. In the absence of a Y chromosome, you make ovaries. You don't see the androgens early in life, and you form a female phenotype. And that the brain is just one more tissue that's listening to this organizing signal. And in the presence of testosterone, the brain will be organized in a masculine fashion. And in the absence of testosterone, it'll get organized in a feminine fashion. And there is a certain, elegance to that. And I won't subject you to all the experiments, but it will, from 1959 on, for the next 40 years, there will be an explosion of experiments where pick a mammalian species, doesn't matter which, and manipulate hormones early in life. Shruti, don't get me in trouble by posting that. What was I saying? Pick a mammal, different species, and pick a behavior, taste aversion, fear conditioning, whatever, and sucrose preference, whatever behavior that shows a sex difference, then manipulate hormones early in life and show that testosterone exposure results in a masculine phenotype for the behavior, or that the absence of testosterone early in life will result in a feminine phenotype in the behavior. And you've got a paper, right? So guinea pigs, mice, ferrets, rats, cats, dogs, and to a remarkable extent, the hypothesis held up. In general, no matter which mammalian species you're dealing with, and no matter which behavior shows a sex difference you're looking at, you can make the animal's behavior as masculine or feminine as you want just by controlling how much androgen they're exposed to early in life. When during early life, right, would be different. For guinea pigs, they got to be exposed prenatally. For rats and mice, postnatals, not, is in time, etc. Everyone get the idea? And that's part of what was made the organizational hypothesis an enormous success. It was a simple verbal equation that explained a lot of data. And I always liken it to the revolution that happened, before I was a scientist, but that you read about, where they're trying to figure out that DNA and RNA, all that, this simple idea that DNA makes RNA and that RNA makes protein. That was a revolutionary idea that happened about 1965. And what made it revolutionary is it explained so much data that was out there, but it was easy to remember. Questions about that? Good.
Is the testosterone like a dating effect where it needs to be like a threshold? Like the degree of casting?
It's a great question in terms of if you wanted to fully masculinize the behavior, when you provided it and how much you provided it mattered and varied by species, and by even within a species would vary for one behavior versus another.
And then like the amount of mounting or the amount of lordosis would like, that's also not gated. It's like you'll see it for more femme mastery.
It's another great question. And I will say again that it depends on the behavior. And for those particular behaviors, the mounting and the lordosis, I'll remind you that it's not enough that you manipulate the hormones early in life, but if you want to gauge how masculine or feminine the brain is, you also have to give activational hormones in adulthood to see how they respond. And so another way to phrase the organizational hypothesis is that exposure to androgens early in life primes the brain to respond to androgen in adulthood, to activate masculine behavior, and to not respond to ovarian hormones in adulthood. Whereas in the absence of testosterone early in life, the brain is not primed as an adult to respond to testosterone much, but is ready to respond to ovarian steroids. Very elegant. In the early days, so that happened in 1959, that this hypothesis was proposed and almost immediately adopted, and everyone went off to the races. You might be surprised to learn that even though they explicitly suggested that the early hormone exposure was going to permanently change the structure of the brain, nobody went looking for sex differences in the brain because they expected that these sex differences would be subtle. But once people did start looking, it turned out that there were parts of the brain where you could look at a microscope slide without a microscope. Just hold up the microscope slide to a light, and you could tell whether that was a male brain or a female brain. And the best studied example of this, and these were called sexual dimorphism. So sexual dimorphism, as you know, is a property of species where one sex has a different structure than the other. The canonical example is the mane on male lions, right? Female lions don't have that mane, but males do. So that's a sexually dimorphic structure. And Darwin worried about why that happened, because the mane's a terrible idea for being a lion, unless you're trying to impress female lions and get them to persuade them to take sperm from you. And so Darwin's second book explained how natural selection would arrange for that to happen. Good. Pardon?
Why is it a terrible idea?
What function does it serve? Right? I mean, if you're eating, right, it gets in the way. It doesn't help you catch anything. If you're fighting, it gives your opponent something to grab onto, right? So this is another class, but it's a sexually selected structure. It's a structure whose only function is to persuade the other sex to let you reproduce with them. And it is a characteristic of sexually selected structures like that once they're in place in a population, they're there forever. Because any female who thinks, well, that male over there with no mane, he's sensitive and he's caring and he's looking after me, I'm going to mate with him. And she can do that, but her sons are going to be at a distinct disadvantage And sooner or later, that sort of female bucking the trend will not gain any genes in the gene pool. Yeah. And I have to tell you, so this is one of my favorite factoids, is that we knew from the paleontology that there had been lions in Europe, right? But we didn't know, whether they were like African lions or like other big cat species. But until they found Neanderthal drawings in European caves where they showed male lions and female lions. And so that's how, the mane itself doesn't leave a fossil, but that's how we know, they were like African lions in that regard. Okay. So the best studied sexual dimorphism in the brain is portrayed here, and it's a portion of the preoptic area that is in the anterior hypothalamus, and it's a portion where there's a nucleus that you see in Nisselstein that's about 3 to 5 times greater in volume in males than it is in females, and so they called it the sexually dimorphic nucleus of the preoptic area, or the SDN-POA. And the SDN-POA conform beautifully to the organizational hypothesis. So normally, males have testosterone early in life and testosterone in adulthood, and they have a large SDN-POA. Females normally aren't exposed to much testosterone in any of those cases, and they have a small SDN-POA. But if you provide a female with testosterone, even a single injection of testosterone, one time in her early development, and we know all the hormones gone within 24 hours, that will permanently alter the structure of her brain, because when she grows up, she'll have a large SDN-POA. And if you wait until the female's grown up and give her extensive treatment with testosterone, it's too late. You've passed the organizational period where you can change the structure of this part of the brain, and so it'll be permanently feminine. And we're not showing it here, but if you castrate males at birth, they'll have a much smaller SDN-POA when they grow up. How's that? Okay. And this is just one of many sexual dimorphisms that were studied. So there became another time when there was a bumper crop where you could look in the brain, and there was a time there, like starting in the late 1970s, where you could have filled a scientific journal with all the reports of sex differences in the brains of choose a species and choose a part of the brain. Most of them were in the hypothalamus, but pretty much anywhere you looked, if you started looking for whether the sexes differed significantly in terms of the structure, you could usually find it. And so there was a heyday of that. And politically, I can tell you that for whatever reason, by then we're like at the third wave of feminism. The people that found these things didn't get much guff from the public. It helped that everyone was studying animals and not humans. But as we'll see, eventually people looked in the human brain and they found sex differences there. And for the first 20 years that happened, nobody thought there was any political consequence of that. What's been interesting to me is for the past 20 years, suddenly now people are concerned that maybe there aren't really sex differences in the human brain. Maybe that's just, another aspect of the patriarchy trying to put women down, right? Which is an important concern. But what's interesting for me is to see how the concern before 1970, if you suggested men and women had different brains, you might have gotten castrated for it, right? But then the 70s and 80s comes and nobody cares, 90s, and now people care again. Yeah. I'll just say one more thing. We will talk about sex differences in the human brain, but let me point out to you that just because there's a sex difference in the human brain, that doesn't mean that hormones had anything to do with it. show me a sex difference in the adult human brain. And I don't know if it got there because of testosterone early in life, or if it got there because boys and girls are treated so differently that it caused their brains to develop differently, right? As neuroscientists, we know that's very important. And it'll be the main theme of what we talk about in the last quarter of this class. But, yeah. Yes.
The preoptic testosterone was developed.
What's in the preoptic area, in rats, we know testosterone is important for this because we can do the experiment. We can provide the rats testosterone early in life and wait till they grow up and see what happens. Is that the question you're asking? For some reason, IRBs won't approve doing that in people. Yeah, good.
You need to be ethical because I bought that social and have seen my father take along. Did my same brain helps me?
Would it be ethical to test?
Whether like socially. I was voting with defensity.
Again, you can't ethically do that. I'm going to take a bunch of XY babies and raise them as females, and see what happens to their brain structure. Right? How will we ever know? It's a great question, and my advice is get used to ambiguity. And I'm glad, right? Because we could answer the question if we didn't have any ethics. Then you'd get a definitive answer. But there you go. And so here's the SDN. In the medial amygdala, there were sex differences. All of this look at sexual dimorphisms. It actually started in songbirds like canaries and zebra finches, where males who sing, that the nuclei that are important for singing are much larger, more prominent in males than they are in females, for example. And we're going to talk some more in a bit about the spinal cord, where there were sex differences. And the theme that all of these sexual dimorphisms in animals share is that you can make the nervous system as masculine or feminine as you want just by controlling androgen. Right? And typically, if you want a fully masculine phenotype, you got to expose the animal to androgens early in life. Good.
Has anyone done research with like finger digit ratios and then postmortem, like scans the brain to see the structural differences?
You're way ahead of me. So the answer is easy, no. But Justin brings up a point. All of these sexual dimorphisms, I mean, eventually people will find an SDN-POA in humans, right? But they're all way too small to see with non-invasive imaging, right? So the SDN-POA in rats or humans, it's about the size of a grain of sand, a biggish grain of sand. There's no MRI machine that can resolve that, and probably never will be. It probably require magnetic fields that would pull the brain apart.
Does a finding the floppy hormone at puberty, does that result in a feminized nervous system?
Andrew, you mustn't ask me about puberty blockers, then I will get arrested.
Seriously.
We may discuss them later. I will just tell you that is the third rail. I probably will lecture about it. I have lectured about it in human sexuality, and each time I'm rolling the dice. Yeah. Sorry. Okay. Questions about that? I need to tell you about one hiccup in this story that applies to rodents, and I want you to know about it because it's important for me understanding the literature, which is, it turns out that in rodents, the masculinizing effect of testosterone on the brain is through estrogen receptors, not androgen receptors. And to understand that, I need you to understand this mechanism that's known sometimes as the aromatization hypothesis. The molecule testosterone can, in a single chemical reaction, be converted to the molecule estradiol. In fact, when the ovary is producing estrogens, it does so by first manufacturing androgens. And some of those androgens go into circulation. But the ovaries are chock full of an enzyme called aromatase. And aromatase converts testosterone to estradiol. And it converts other androgens to other estrogens. Everyone get that so far? So in rodents, when early testosterone masculinizes the brain, the testes are making testosterone, it gets to the brain, and inside brain cells in the hypothalamus and elsewhere, it is converted by aromatase into estrogen. And then the estrogen binds not to androgen receptors, but to estrogen receptors. And in the rodent species, that's the step that triggers masculinization of the brain, permanent masculinization of the brain in terms of structure and function. And it's confusing because we're used to thinking of estrogens as being feminine hormones, but natural selection doesn't care whether you're confused, and natural selection pays no attention to labels we give it. So estrogens are not female hormones. They are hormones that are more prominent in females than in males. And testosterone is not a male hormone. It's a hormone that's in higher levels in males than in females. But both hormones play crucial roles. No, that's not true. Both hormones play roles in both sexes. Estrogen, in fact, is crucial for development. Interfere with estrogen signaling, and the embryo is doomed. You can live without androgen receptors, but in typical females, androgen receptor and testosterone also plays a role. Good. Maria?
So females don't necessarily have estrogen signaling until later on in life.
Pardon?
Females don't have estrogen signaling until after development.
Are you talking about female humans? Yeah, I mean, here's one of the paradoxes that we have to talk about when you talk about this hypothesis, is pregnant women are making estrogen like mad all the time. And so every fetus is exposed to high levels of estrogen early in life.
Then why did estrogen masculine?
So you've jumped ahead. So in rats, how come females aren't masculinized by the maternal estrogens? And the answer is because in fetal rodents, there's a protein in circulation known as alpha-fetoprotein. Humans have an alpha-fetoprotein too. It's called that because it was the first fetal protein that was identified. Okay. In rodents, but not people, in rodents, alpha-fetoprotein binds to estrogen, but not to testosterone. So I should have paid you to ask me that question. So normally it's estrogen binding estrogen receptor that alters gene expression and masculinizes the brain. Any estrogen from mom is sequestered by alpha-fetoprotein in circulation. And it just so happens that alpha-fetoprotein binds to estrogen, but doesn't bind to testosterone. So the males making their testosterone, they get by the alpha-fetoprotein blockade and go to the brain and get converted to estrogens inside brain cells away from alpha-fetoprotein, which is a plasma protein, and do the dirty deed.
You know what you deal with female and adults with testosterone? Is that maximize their nervous?
In rats, if you give a developing female testosterone, is that what you asked?
An adult.
If you give her testosterone, it's a fetal protein. Alpha-fetoprotein is gone by then, so it's not going to play a role. But yeah, it'll get to the brain. It will have effects, but in animal models, at least, you won't be able to permanently affect the structure of the brain. It's too late.
Testosterone the brain goes back to its adult state.
Testosterone goes to the brain?
Stop providing testosterone, it goes back to its original state.
The brain goes back. No. If manipulating hormones in adulthood is too late to change the structure of the brain. They have to be exposed to testosterone early in life to have organizational effects. And we talk about these things ad nauseum in sexual behavior, human sexuality class. Questions about that? And by the way, the aromatization hypothesis, you have to understand it to understand the rodent literature, but it almost certainly plays no role in human, right? So if there are any effects of hormones early in life, masculinizing the human brain, they can't be through estrogens. Because remember the AIS women? Nothing wrong with their estrogen receptors. They got plenty of estrogen, but they're very feminine in their behavior. So if there's any hormone that acts early in life to masculinize human behavior, it's got to be working through the androgen receptor. And there may not be one, right? It may be that socialization alone explains human sex differences in behavior. Okay, I beat you to it. Okay, I saw you coming. So the other model that I talk about a lot because I was involved in it is in the spinal cord, where again, you can take a section of spinal cord, and if it's from the lower lumbar spinal cord, you don't need a microscope. Just hold it up to a light. And if you see these two clusters of dark motor neurons in the ventral horn, right at the midline, then you know it's a male. And if you don't see them, then it's a female. So these are motor neurons, and they innervate muscles that are called the bulbocavernosus muscle. And so the nucleus is named the spinal nucleus of the bulbocavernosus. And it is totally under the control of androgen receptor, not estrogen receptor. So in the spinal cord, the aromatization hypothesis does not hold. So for example, you can look at rats that have a defective androgen receptor, and they are fully feminized in terms of the SNB. One reason I talk about this in neural development is because we understand the site of action for testosterone to cause this. It turns out that when testosterone, and this is why this material used to be in the chapter on apoptosis. It turns out that early in life, testosterone prevents these motor neurons from dying. If you look at a female rat on the day before birth, she has bulbocavernosus muscles wrapped around her clitoris. She has motor neurons that innervate that muscle. The synapse between motor neurons and muscles works. So the neuromuscular junction is functional. But in female rats, starting on the day of birth, both those structures die. Both the muscles and the motor neurons die. In males, they stick around. If, on the day of birth, you give a female rat a single injection of testosterone, then you stop the death of the motor neurons and the death of the muscles, and she will have them forever. So just in this case, we understand why there's an organizational effect of testosterone. You have to give the testosterone before the system dies. If you wait until after the muscles and motor neurons have died, it's too late to save them. And we know that when that happens, the testosterone is acting not on the motor neurons, but on the muscles. So the testosterone keeps the muscles alive, and if the muscles survive, then they keep the motor neurons alive. And there's pretty good evidence that when testosterone acts on the muscle, it causes it to provide some sort of trophic factor to keep the motor neurons alive. And we're all quite used to that concept, having talked about apoptosis and the neurotrophic model of controlling cell death. Questions about any of that? I will tell you that when I named the SNB, I tried to think of a way to make it the SMB because those are my initials, and I just couldn't think of a name that didn't, strain credulity. So you're stuck with it. Questions about that? Oh, and here's the study that was done to show that. So you could take females who are carrying the androgen receptor knockout on one X chromosome, and because of you can look at females who are mosaic for whether they have a functional copy of the androgen receptor gene or a dysfunctional copy of the androgen receptor gene. You give them testosterone early in life to keep the system alive, and then you ask, was it only the motor neurons that have an androgen receptor gene that were spared? If so, that would suggest that androgen acts on the motor neuron itself to keep it alive. Everyone see the logic of that? But it turned out that motor neurons with dysfunctional androgen receptor genes were just as likely to be spared as motor neurons with functional androgen receptor genes. So the sparing of motor neurons is not a cell autonomous response to testosterone. It's not the motor neuron itself that's responding to the hormone signal. Rather, the hormone signal is acting somewhere else, and then some other structure is instructing the motor neurons to stay alive. How's that? So for lots of animal models where sexual dimorphism has been found, it turns out that hormone is modulating the amount of apoptosis. And we're not going to talk about lots of these areas, but we've talked about the SDN-POA and the SNB. In both of those cases, testosterone prevents cell death and results in males having more neurons than females. But there's another hypothalamic nucleus, the anterior ventral paraventricular nucleus. I suggest you just remember AVPV, and I do want you to remember that. It turns out females have more neurons there than males do. And in that case, testosterone promotes apoptosis. So far of those animal models where there's sexual dimorphism in the brain that involve differences in neuron number, so far in all of those cases, testosterone is regulating how many cells live or die. And one way that people have shown that is you can get animals that overexpress BCL2, remember that, the anti-apoptotic protein. If they overexpress BCL2, then there's almost no neuron death. And when you do that, the sexual dimorphism doesn't appear. How's that? Or, I don't show it there. Oh, I do have it. Or you can knock out the preoptic gene, BAX, so that there's lots of death. And again, in that case, there's no sex difference in the number of neurons that survive. Just to keep us all on edge, imaging studies have looked at whether there are sex differences in the human brain. First of all, there's a sex difference in the size of the human brain, right? Men tend to have bigger bodies, and in general, the size of the brain correlates with the size of the body. So, unsurprisingly, male brains tend to be heavier than female brains. You're sophisticated enough as neuroscientists not to fall into the trap of thinking, well, if the brain is bigger, it must work better, right? Sorry, things aren't that simple. And the other thing that I wanted to get across is that people have tried to do like meta-analyses, looking across lots and lots of different studies. And there are certain places where there seems to be a sex difference. This scale is one in which yellow areas are ones where the structures tend to be bigger in males than in females, and light blue is when they tend to be greater in females than in males. And so there are certain areas that do seem to be larger in men than in women, and other areas that tend to be larger in women than in men. And as you read the literature, you can find people talking about the various parts of the brain where these sex differences are found and stating that they explain sex differences in behavior. And I'm totally skeptical about any of that. The other thing I want you to understand is we don't know how these sex differences in the brain came about. So there are no easy animal models that we know mirror these particular brain structures, because this is all in the cortex, right? So we can't look to them. And for all we know, these sex differences in the human brain are not due to hormones at all, but are due to the fact that we socialize girls and boys differently, and therefore that could be changing the structure of their brain. Because as we all know, as neuroscientists, experience alters the structure of the brain. Otherwise, there'd be nothing to study in learning and memory studies, right? Questions about that? Okay, so let's have a Kahoot. We're going to talk about some other things, and Justin tried to steal my thunder. So from all of this, we can ask the question whether prenatal steroids also masculinize human behavior. Can't look at brain structure because we talked about the problems we talked about like that. And now we get to, yeah, experiments that have kind of come to define my research career for reasons that make no sense whatsoever. Okay, so until 2000, I was very happy studying the role of hormones in altering the development of the nervous system in rats and mice, and sometimes guinea pigs and dogs and humans, vaguely. And every time I wrote an NIH grant, I always said, these studies might be relevant to human health one day. But I never actually believed that. And because I don't actually care. I mean, the only reason I ever did science is because I was curious, right? But you got to say that it's relevant to humans, or else they won't give you the money. And don't worry, when I said it was relevant to humans, nobody believed me anyway. And so except for looking at post-mortem materials of the spinal cord, I never worked with humans. I'm in my office and I read this paper that says that there's a sex difference in the ratio of human fingers that's present in 2-year-old humans. What? I'd spent my entire adult life studying sex differences. I'd never heard of this. What are you talking about? And like most people, I looked at my hand to see, what are they talking about? But I knew that if it's present in 2-year-old children, I mean, given what you know about sexual differentiation, it's almost certainly a reflection of prenatal testosterone, right? Not likely to be anti-Müllerian hormone, right? So, wow. And I happened to be at Berkeley at the time. It's like, I'm in the Bay Area, and there are loads of gay people. Maybe we should look at this. So it's pretty interesting. The German anthropologist who first talked about this in the late 1800s, he said, when you ask people questions, there's some questions everybody gives you the same answer to. How many legs does a spider have? Everybody knows the answer to that. And yet, how many legs do crabs have? Well, you get different answer. People aren't sure about that. And he said, then when I ask people, which is longer, your ring finger or index finger? He said, every time I ask someone that, nobody gives me an answer. They all look at their hand and then tell me whatever they see there. And I've noticed that women are more likely to say they're equal. And men almost always say the index finger is shorter. And there's a controversy, and people are looking at primates. But by the 1930s, someone actually takes, a sample and shows that there is a sex difference there. So and then it gets forgotten, as far as I can tell, in the literature until 1999. Anyway, so here's a typical male hand where we can take a Xerox of people's hands, line it up on a ruler. And so this index finger which is the second digit, is 78 millimeters. The ring finger is 83.5. We can take a ratio of that, and it's about 0.934, always close to 1. Or here's a female digit ratio, female hand, where the index finger is longer than the ring finger, right? And so that difference is larger. So in general, women have a larger 2D, 4D than men. Now, I see all of you looking at your right hands. Stay calm. You're going to be fine. I'll talk you through this. Notice that in both these cases, these are the right hand. Some people report finding sex differences in the digit ratio of the left hand. I've never seen it. And we've published, 4 or 5 studies at least, looked at thousands of hands, and I've just never seen it say. So for some reason, if this is responding to prenatal testosterone, for some reason the right hand is more sensitive than the left, and I have no understanding why. I think that the whole thing is just kind of an accident, sort of a side effect of other things. It's of no functional significance. Anyway, so the difference is it's a medium-sized difference. So one way of asking how big a difference is to ask how many standard deviations are different between the two population means, right? And in psychology, a D prime of half is considered medium. And Cohen, the guy who proposed this stat of D prime, he called that a medium effect. And I love the example he gave to it. It's equivalent to the difference in height between 14-year-old girls and 18-year-old girls, right? So we all know there's some 14-year-old girls that are taller than some 18-year-olds, right? What I love about that is you can imagine, what if I took 10 randomly selected 14-year-olds and 18-year-olds and put them in a group and mix them together? If I ask you to tell who's 14 and who's 18 based on height alone, you'd make lots of mistakes, right? You wouldn't be, sometimes you'd classify a tall 14-year-old as being 18 when she isn't. Everyone get that? You can sort of visualize that. On the other hand, if I separate them and I tell you one of these groups is 14-year-old and the other's 18-year-old, you'll know which is which and you'll be right 99 times out of 100. If they're randomly selected groups of 14 and 18-year-old girls, right? So it doesn't tell you anything about an individual girl, but when you compare groups, it can importantly tell you which groups are different from one another. And so we're in the Bay Area, and we go to street fairs, and we ask people to tell us, who are you sexually attracted to? Who have you had sex with? Who would you like to have sex with? And several other questions. And then we also ask at the end, we put in 2 questions that were really meant there to kind of fool people. We said, well, which hand do you throw with and which hand do you write with? Because now our next question is, can we please Xerox your hands? And what I loved about Xeroxing hands is, they're easy to measure blind, right? I don't, you just, and the ruler's right there. How hard can it be? It was an anonymous survey, but if you want to know how we got people to answer all these personal questions to strangers, we offered them a prize, a $1 lottery scratcher ticket. People will tell you anything for a $1 lottery scratcher ticket. It was a pretty awkward moment when I went to 7-Eleven that morning and said, I'd like, please, 800 $1 lottery scratcher tickets, and can I have a receipt, please? And I had to pay in cash, of course. So we do this, and on the left hand, we didn't see any sex difference in digit ratio, right? On the right hand, we did. We saw that heterosexual women had larger digit ratios than heterosexual men, as we expected. And then we found that straight women had more feminine digit ratios than lesbians. That in fact, lesbians had digit ratios that were as masculine as those of males in our sample. And, well, that seemed strange. So we published that in Nature, and never in my life have I gotten more emails. Email was a new thing, letters, phone calls, phone messages from utter strangers in my life, The office was kind of inundated with them. And they were of two kinds, right? One kind was, I know you're lying, you're gay, and you're lying about these data, and you're doing that to justify the gay agenda. Hopefully none of you have heard of the gay agenda, right? But there was a thing in 2000, 2001. And the other were everyone who and some people, it was, my child is gay, thank God you proved that it's not his or her fault. And others were, everyone who identified as gay was very supportive, right? But it was a very interesting situation. So we'll talk about that some more when we get back. So remember, before the third midterm, we're also going to talk about the interlude about the empiricists. So see you next week. It is time, isn't it? Oh, wait. Oh, I got more time. Stop. Stay. I got to use this. So we went back the next year. We went back and did the same thing, only we only went to the Oakland gay street fair. Oakland is a great place to find lesbians. It's just full of them. And I just want to point out to you, let me go back to say that we saw no difference between gay men and straight men, right? So straight and gay, we didn't see any difference between them. Come back to that later. We go back the next year and we do the same thing, but we ask them one more question. Do you consider yourself butch or femme? And we didn't define it. And eventually we got like nearly 200 women who were willing to answer that question. And indeed, on the right hand only, the women who identified as butch had more masculine digit ratios than those who identified as femme. And what happens next, I look back on this and I think I was very lucky in some ways, is because what happens next, we published that in 2001. No, I guess in 2000. And for a while, every calendar year thereafter, somebody else published a replication. So I'll never do a study in my life that will get replicated where anyone would bother to replicate. Than this. In part because it's so easy to do, right? You don't need a cyclotron. You just need a Xerox machine or a ruler, a nice plastic one that's clear and it's got millimeter markings on it, right? The equipment costs are pretty low. And so far it's been replicated a lot. My favorite was this study that had the smallest sample size, where they compared 7 pairs of monozygotic twins, girls, women, in which one was gay and the other was straight. And lo and behold, on the right hand, the lesbian twin had a more masculine digit ratio than her straight twin.
Two questions. What does butch mean?
Well, what does butch mean? Butch means in LGBTQ circles, a butch lesbian feels more masculine or acts more masculine than a femme lesbian. And so, sorry.
Question mark, why is the name not used?
Why is my name? Well, because you mean, these are the replications of the study with my name. Yeah. I do love seeing my name, but it's not. That seems like cheating. Anyway, back to this. So on the east coast of America, they found 7 pairs of monozygotic twins. That was later replicated in a Japanese sample, and then even more recently replicated in a European sample. So it seems like, wherever you find monozygotic twins who were discordant for sexual orientation. Now, other questions about that? So I'll finish here just by saying I did get some guff for this. So this is from Salon, and I don't know who put this graphic together. There's not a bad likeness, scientists, you'll wear glasses, tie and lab coat. No, no, that's never happened. But there were two lesbian columnists. Here's one of them who talked about the study, and this particular woman said she's reading the headline in the newspaper and she's really angry about it. And she says, why would anyone do a study like this? She said that it was mean-spirited science. That it was mean-spirited. Twice she says it's mean-spirited to do it. And, it's like, well, are there some questions that scientists should never ask? Something like that. And I will also tell you that for the first time in my life, when reporters would call me, they wanted to know what my sexual orientation was. Well, why are you asking? Well, because if you're gay, maybe you're biased in this. And I said, well, and I refused to answer because they said, well, if I say I'm gay, then you're going to accuse me of pushing the gay agenda. And if I say I'm straight, then it makes it sound like I'm trying to distance myself from gay people. So I'm not going to answer that question. But I will tell you that the research team involved the usual mix of sexual orientations that you'd expect in the Bay Area, right? So, okay. So, yeah. So when we get back Thursday, then we'll talk some more about digit ratios and sex and stuff like that.